Hello to everyone. I am Darius Kavaliauskas and my colleague Gregor Božič. We are going to present for you today guidelines for conducting forest genetic monitoring of the 7 tree species within the project LIFEGENMON. As you can see, those 7 tree species or group of species are Abies alba, Abies borisii-regis, Fagus sylvatica, Fraxinus excelsior, Pinus nigra, Populus nigra, Prunus avium, Quercus petraea, and Quercus robur complex. So overall in this presentation, we are going to overview the species selection issues for forest genetic monitoring. Then we are going to talk about the preparation of species of specific forest genetic monitoring guidelines for those 7 target tree species or complex of the species, which differs in their biology and distribution. Then after all, we are going to introduce you to some details about the field work and plot establishment procedures and those guidelines, and then we will give you some examples from several guidelines. For example, we will look a bit more in detail in Abies alba verifiers and the FGM forest genetic monitoring plot selection of Populus nigra. So main work on the guidelines started a bit later than the project. It started in 2018, and since the first monitoring plots and first results was collected for Abies alba and Fagus sylvatica. So one of the most important issues within forest genetic monitoring is the target tree species selection or group of the species selection. Therefore, in 1996, Namukung et al. Was the first who was trying to prioritize species for forest genetic monitoring on different characteristics, for example, on economic significance or ecologic significance of the species, if the species is rare or endangered, indicate, or if the species represents huge ecological region, or it's really important and indicative. And as you can see, for example, in this map, which shows the dominant species 1 by 1 kilometer of the Europe. There are a number of species which are dominant in different regions, therefore they can be in focus for forest genetic monitoring. Later on, Namukung in 2002 improved and supplemented the characteristics for species selections in order of importance. So for example, he used 8 characteristics like if species is harvested or no, if species are rare or scattered, if the reproduction system is specific, and if species is dioecious, or if it is keystone species, or there are some specifics in pollination system. However, these suggested characteristics are relativized by the statement that it is not necessary for all candidate species to meet all of those requirements, those characteristics. Additionally, it is not fixed how many of them have to be fulfilled. But despite all these negative, let's say, points, those characteristics are still useful for preliminary selection of species for forest genetic monitoring. As the importance of species is different in different regions, as we saw in the previous map, the target species can vary according to the region and location. Another thing what was underlined by Namukung, that the discussion with relevant foresters and stakeholders are really important during the selection process of the species for genetic monitoring. So in 2004, a German expert group on forest genetics proposed a scheme for species selection and suitable for forest genetic monitoring in the Federal Republic of Germany. This scheme is based on 9 criteria for prioritization of species together with evaluation scale. So as you can see, there's 9 main criteria is like risk of extinction, if it is high or there is no extinction risk, rarity of the species, economic significance of the species, for example, manner of pollination, this wind or insect pollinated species, and if there are availability of primary data from another different type of monitoring programs, or maybe there are available data, genetic data already, or genetic markers which can be used for genetic monitoring, which makes the genetic monitoring, let's say, a bit easier. So based on this scale, on this, based on this prioritization, the German expert group selected, let's say, 4 groups of trees: 2 dioecious tree species, 2 coniferous tree species, 2 insect-pollinated tree species, and 2 relic tree species, and concluded that those species should be used for genetic monitoring. So, like 2 dioecious is European beech and oak, 2 coniferous are silver fir and Norway spruce, 2 insect-pollinated would be small-leaved lime and wild cherry, and 2 relic tree species like black poplar and elm. So in 2006 in Germany was implemented pilot study for genetic monitoring for European beech and wild cherry. And then in 2016, the project GenMon started a country-wide genetic monitoring program on European beech and Norway spruce, and therefore over 20 monitoring plots for these 2 species were established in Germany. Also, again, working group on forest genetic monitoring offered, let's say, the workflow how to be started forest genetic monitoring, and of course the main and important step is the prioritization of species for genetic monitoring, and then it follows identification of genetic monitoring regions for selected species, selection of genetic monitoring regions, units within the regions, and then of course establishment of plot, and then selection of indicators and verifiers. But let's go back to the prioritization of species. So therefore, again, working group used 6 criteria for species selection, and for example, there was such criteria like species which having both microsatellites and SMPs information available, or species which are distributed in broad ecological categories, wide or restricted distribution, and for example, stand-forming or scattered tree species should be, or species that present genetic conservation units with predominantly 50 trees, or species, for example, which are marginal or rare, but with a minimum number of 50 trees, or also species which is important maybe in the light of climate change in southern distribution, and also species which was used in relevant projects on forest genetic monitoring maybe can be used for species for genetic monitoring. So therefore again, evaluated 15 tree species, and out of those, they selected several for genetic monitoring. So this is an overview of 7 LIFEGENMON target tree species, and the assessment were based on all previous mentioned characteristics. And for example, as you can see, we selected 7 tree species, which are all, of course, ecologically important, but for example, only 2 scattered tree species, it's Populus, nigra, and Prunus avium. Also, we have 2 conifer tree species, it's Abies and Pinus nigra, and 1 insect-pollinated species like Prunus avium, and 1 dioecious tree species, for example, Populus nigra. So one of the aims of the project LIFEGENMON was to prepare forest genetic monitoring guidelines for these 2 model tree species, for which we established actual monitoring sites in the transects from south of Germany to north of Greece. For Abies, alba, and for Fagus sylvatica was established 6 monitoring plots, and for additional 5 forestry species, guidelines were prepared based on expert knowledge. For implementation, of course, forest genetic monitoring at national and regional and EU scale. So all those guidelines for those 7 tree species contained biology of the species, distribution of the species, economic and ecological value. Those guidelines also gives a guidance on how to select and establish and maintain forest genetic monitoring plots, and how to record all field level verifiers and background information for different model tree species at basic, standard, and advanced genetic monitoring levels. So So overall guidelines of our 7.3 species contains, as you can see, executive summary, species description, species reproduction, environment, and threats. Also, it contains plot establishment and maintenance procedures, how to select, install the project in the plot in the field, how to label the trees, how to sample, and how to maintain the plot. And also, it describes how to record verifiers and background information at the 3 levels of genetic monitoring. And also, it gives the description on how to record background information like DBH class distribution, high class distribution, bud break, and senescence. Overall, in LIFEGENMON project, we used 3 indicators and 20 verifiers, but into guidelines falls only the first verifiers from indicator selection. It is mortality and survival, flowering, fructification, and natural regeneration. Those 4 verifiers falls directly into guidelines and are assessed directly in a forest genetic monitoring plot. This is an example of overview of verifiers and background information for Abies alba and Abies borisii-regis. All these 4 verifiers are described for basic, standard, and advanced levels. And as an example, fructification, we can see that it should be assessed at the stand level every year, at the basic level every year, on the stand level, at the standard level, it should be recorded on individual tree level. In the same year of the flower, when it is assessed flowering, and for example, at advanced forest genetic monitoring level, it should be recorded fructification by counting fruits during the same year of flowering, of course, in a specific time frame. So from here, I would like to give a floor to my colleague Gregor Božič to continue this presentation.
I will present highlights of LIFEGENMON species-based verifiers and background information. To provide forest genetic monitoring, we selected 4 common verifiers for all 7 tree species. And these are mortality, survival, flowering, fructification, and natural regeneration evidence. Very important is also background information. DBH class distribution, height class distribution, flowering synchronization. Sex ratio is also important for species which has male and female flowers on different plants, while crown dieback is important for Fraxinus species. Different species have different characteristics. Unique genetic scheme is not possible for all species. For stand-forming species, regular plot design is used, while for scattered tree species, the specific forest genetic plot design are developed. For example, for Populus nigra, we follow the meta-population concept with local populations along the river system, while for Prunus avium, it's very important that forest genetic plots are in distance of 8 to 10 kilometers from cultivation of domesticated cherries. The guidelines for Fagus sylvatica and Abies alba and Abies borisii-regis were prepared based on the results from actual test sites. 6 plots were established in 3 countries. The plots were established in the transect from South Germany to Slovenia and to Greece. And all measurements were done in these sites, and all results are related. These test sites will remain activated as a permanent forest monitoring plots for a long-term observations. In these slides, you can see the regular forest genetic monitoring plot scheme for stand-forming species. How to select the plot? We consulted with the local foresters. We use also orthophoto pictures and also did field surveys. Plot for forest genetic monitoring includes 50 major reproducing trees, which are distributed over the forest stand with a distance of 30 meters between selected trees. It's very important that the tree plot is selected in the area with a sufficient density of natural regeneration. The establishment of natural regeneration subplots is carried out during germination following a strong or massive fructification event. For scattered forestry species, special forest genetic monitoring designs were developed. Hereby, I will shortly present forest genetic monitoring design for Populus nigra, which is following the metapopulation concept. European black poplar is an ecologically important pioneer forestry species of mixed riparian ecosystems. Major and offspring subpopulations are not overlapping, and Populus nigra naturally forms a metapopulation of interlinked local population, as you see also here in the picture. Populus nigra metapopulation is mainly exposed to high hybridization pressure because of plantations with exotic poplars, especially with Populus deltoides or Populus with hybrid poplars with with Populus deltoides and hybrid poplars, which were from plantations. Plantations were established on the alluvial sites, and therefore it's a strong pressure to the metapopulation for hybridization. In this way, because of this, we need to do genetic identification of taxonomies Populus nigra trees. So we need to do genetic identification for Populus nigra trees. And we have to do it also by use of molecular markers. So genetic monitoring plot should be established in interlinked local populations which represent metapopulation parts along the river system. For a genetic monitoring plot represents a group of reproducing pure Populus nigra trees of different genotypes selected in local populations along the river system. Number of selected trees in each local population depends on its population size. If you have local population with more Populus nigra trees, then it's good to select more Populus nigra trees from this population. Usually, local populations consist of 10 to 30 trees, which are occurring in 1 to 10 hectares. So we suggest to select Populus nigra trees in local populations in that way that we should do monitoring at least to 20 trees with maximum distance of 5 kilometers per one local population. Genetic monitoring plot have to be representative, and it should consist 50 reproducing pure Populus nigra trees distributed over a metapopulation area. Ideally is that trees should be selected in sex ratio 1:1 of male and female trees. So to ensure representative sampling across the metapopulation, it's important to design a genetic monitoring system with randomly selected reproducing trees in local populations along the river system and monitoring subplots of natural generation centers in both sides of the river in the given area. For a genetic monitoring of scattered tree species like European black poplar, in contrast to stand-forming tree species like European beech or Sirwell fir, is more complex, challenging, and time-consuming, and more expensive. More about Populus nigra forest genetic monitoring will be presented in a poster presentation. For the conclusion, the forest genetic guidelines of the 7 selected tree species are expected to contribute to the establishment of a network of genetic monitoring plots across Europe. There are several activities ongoing in a number of EU countries on forest genetic implementation, that is in Germany, in Greece, in Slovenia, in Hungary, in France, and in Lithuania. And so we think that forest genetic manual guidelines prepared by LIFEGENMON team are essential. Also, coordination on forest genetic sites and species on the European level is leading by EUFORGENE. Thank you very much.